Hello and welcome to week 3 in biopsychology. In our Zoom meetings, and if I do these videos, what I intend on doing each week is covering some really basic content that's in the course book. It's meant to be read. It's not meant to be, done in a presentation, but cover the material in the course book, and then review any labs that we're going to have. That will be the content here. So let's get right into the course book. We're going to be looking first at Chapter 3 and then Chapter 4, starting with the synapse. And in the attention section, I get a little bit into reflexes, and it's kind of getting into the notion of just how fast this system can be. And so what we're going to be learning about in this particular chapter is the process by which communication happens in the presynaptic neuron and postsynaptic neuron. Discuss the public perception of the impact of hormones, and describe the chemical and synaptic effect of a variety of recreational drugs. So those are going to be aligned with the particular assessments that go along with this chapter. So let's talk about the synapses. And here's a little picture in the course book. We have the presynaptic neuron and the postsynaptic neuron, and the direction of the signal is going from left to right in this particular. So the presynapse neuron terminal button, and then the gap, they call the synaptic gap, and then the postsynaptic or receiving neuron on the other side. And essentially what we're looking at is that as the electrical signal travels down the presynaptic neuron and gets to that terminal button, the biochemical status of that particular part of the neuron begins to change because of that electrical activity, and these vesicles inside of the neuron, which are full of a chemical that we call neurotransmitters, begin to migrate actually physically to the edge, to the membrane of the cell. They bond with that membrane and then release their package. They release their neurotransmitter into the synaptic gap. Those enter into that space. They float across, and they bind to receptor areas on the postsynaptic neuron, and thus begin the process of opening up ion channels and creating the electrical potential on the postsynaptic neuron, and the transmission of information goes from one cell to the next. So each one of these processes, pretty amazing considering how fast this all happens. That's why the book begins with some reflexes, looking at that, about how fast this happens in every space, every neuron connecting to a neuron. This bioelectrical chemical process is what's going on. So there's a couple movies for you to watch about, what it looks like, and some animations, some of the neurotransmitters that are out there, the amino acids, monoamines, acetylcholine, neuropeptides, purines, and certain gases like nitric oxide. So we go into one of those, and we go into some detail in that with some movies here. We have Whoa, lost my way a little bit. Let me regroup here. So we're going to talk a little bit, sorry about that, about the activating receptors on the postsynaptic neuron. These are the receptors of those neurochemicals that are coming in from the presynaptic neuron, having been released from the vesicles. There are two ways that neurotransmitters affect the postsynaptic neuron. The first is ionotropic effects. These are brief on and off interactions that open up the sodium and potassium channels and effectively transmit that information from one cell to the other. The metapotropic effects are longer lasting and slower in process. So sometimes we can realize these processes when we think about, let's say, the speed of particular medications, or how much blood level a particular medication has to have in order to have its psychotropic effect. Drugs that are taken that have immediate effects have mostly ionotropic effect, and drugs that we have to keep in our system and activate much slower on a much more lengthy basis. We might start taking a medication and it's a while before it takes effect. Those will have the metabotropic effect. So one of the ways that we realize this in real life. The next thing that I get into is what happens to these neurotransmitters when they're all used up. And what will often happen is the neurotransmitter will bind to the receptor site, and as soon as that happens, another mechanism actually breaks that bond, and the neurotransmitter floats back into that synaptic gap. Number of things can happen. They can simply diffuse away. They can reactivate another receptor. They can be reuptaked into the presynaptic neuron and recycled, nice little recycling system built into our nervous system. And then they can be destroyed essentially by the presence of enzymes that basically kind of act, they kind of cut the, or break up the shape of the molecule. And that shape of the molecule is really important because the way these neurotransmitters work is the neurotransmitters that are released from the presynaptic neuron are of a particular shape, meaning their biochemical shape, the carbons and the oxygens and whatever other components there are of that molecule compose into a particular shape. And the receptor acts very much like a lock that is unlocked by this particular key because of the shape of the key. And so that's how they work. So what the enzymes do is they either break particular aspects of the chemical off, or they bind to them so the key is now disfigured and no longer has the same ability to activate the postsynaptic neuron. Drugs that we use recreationally mimic neurotransmitters. So hallucinogenic drugs, for example, like LSD, resemble serotonin, and they bind to serotonin receptors and thus produce the effect that they have. Nicotine binds to a type of acetylcholine receptor, and we get an increase in the release of dopamine into the body. And opiate drugs bind to the same receptors as endorphins, very, very giving the unique pain-killing quality of opiate drugs. And so we can see that, part of what I'm going to have you do is to look at the actual mechanism of certain drugs and the reason why people take drugs, and sometimes the reason why they become addicted to them is because they mimic naturally occurring drugs that do the same thing but do it in a more functional, normal way. When we ingest opiates, for example, we're putting more endorphins into our body, but at an abnormal rate that can then become addictive. And the particular quality about chemical addiction to certain drugs is that as soon as you're taking enough of the, let's say, of the external source of the drug, the internal mechanisms for producing those drugs begin to shut down. There's enough in the body, the brain doesn't have to produce its own chemicals, and then we become physically dependent on that particular drug. In addition to that, we talk a little bit about hormones. We have this whole list of hormones that you can kind of review, and we're now into the assessment. The assessments consist of a discussion where I'm asking you to go out there, find an article, find something having to do with hormones, and discuss that in terms of the public perception of hormones and what they really are. And then a four-question quiz where I want you to describe in a single sentence the ways in which information is communicated from the presynaptic neuron to the postsynaptic neuron. And then I want you to research 3 drugs, marijuana, alcohol, and methadone, and describe the biochemistry. Don't describe the effect of the drug. Don't make me high or hallucinogenic or anything like that. Describe the biochemistry. What is happening at the neuron level? This is going to be a little bit deeper research than your first Google search. Feel free to use the web, get a good reliable source, and look up those particular drugs. So there's chapter 3 for you. So we enter now into chapter 4, and we move away from the tiny part, the tiniest part of the nervous system, and we start looking at the larger aspects of the nervous system, the anatomy of the nervous system, made up of the spinal cord, brainstem, and brain, which is the central nervous system, and all the rest of the nerves that make up the peripheral nervous system. Now, I start off with an interesting attention getter here. I think it's interesting, is one of the only Nobel Prizes that was awarded to a person that worked in the field of psychology was actually given to Antonio Moniz for his development of the procedure that we know of today as the prefrontal lobotomy. Now, in an effort to help individuals who were incredibly disabled by their most likely schizophrenia and other types of disorders of that kind, he developed the notion that if we were able to surgically separate the prefrontal cortex from other parts of the brain, that it would lessen those particular symptoms. So he created a device that was essentially a spike about that long that he inserted behind the eye, and with a mallet could punch it through the cranial bones that are at the back of the eye, which are fairly slim, stick it in there and just twirl it around and mush up the brain a little bit. Archaic as it sounds, it was state-of-the-art back in this time, and produced, I guess there's some documentation of success, but over time the negativities of the procedure far outweighed the positives. And essentially, the invention of medications saved the future generations from having to put up with this kind of archaic brain surgery. So this person won a Nobel Prize for that. Now, the sad thing, the really sad thing, is that he was a great inventor of something else, and that is the dye and the procedure used when you inject a dye and the dye goes into the vascular system of the brain so that you can x-ray the vascular system of the brain. Now, that is a system that we still use today. It is miraculous, used for diagnosing blockages and strokes and any other sort of abnormalities in the circulation of blood in the brain. And he didn't get an award for that, yet that is probably the most significant effect that he had on the world of psychology. So in terms of our learning outcomes, we're going to be looking at the difference between white matter and gray matter and why it's important in the spinal cord. I want you to create a presentation as your assignment here of the structural and functional parts of the brain, and we're going to be discussing the moral and ethical implications of using animals for scientific research. So let's get on with the content here. The nervous system, as I said, you can read it, made up of the central nervous system and the peripheral nervous system, the central nervous system, the brainstem and spinal cord, and all the peripheral nerves, peripheral nervous system. When we think about neurons and the speed at which they need to transmit information, this is where we talk about the difference between white matter and gray matter. And let me explain that a little bit. Gray is the natural color of the nervous system. White matter is the nervous system that has been myelinated. And what myelin is, it's a cholesterol-based coating that begins to grow on the outside of neurons, particularly along the axons, the longest part. And it increases the speed of the nervous system. So places in which we need the nervous system to be fast, basically in the peripheral nervous system and in the spinal cord, it's white because it's been myelinated. In the brain, however, the brain parts are so close together and so packed together that there's not as much need for speed, so the brain tends to be that gray color, and the white tends to be the peripheral nervous system. Now, because we need to move neurons fast. Now, there is one part of the brain that is very myelinated, and that is the corpus callosum, and that is the connective tissue between the right and left hemisphere, is white when you look at it, and highly myelinated to increase the speed of transmission from one side of the brain to the next. here we see a graphic in there about the spinal cord and where the protective casing that we call the vertebra or the spinal column. And an interesting thing about this particular graphic is it labels all of the vertebra in the spine, and you can see on the right it's a C1, C2, C3, then T1, T2, T3, all the way down in L and S. And what that stands for is cranial, thoracic, lumbar, and sacral. And you may encounter somebody saying T8 or something when they're referring to an individual who's had a spinal cord injury. They usually designate how high up on the spine did the actual injury happen. And that determines to a degree the level of disability. The higher, the more severe, because functions of the body are, the communication from the brain to the rest of the body is distributed down the spinal column. So up toward the top, we're controlling our mouth and our shoulders and our arms and our hands, and then lower down, we're looking at our trunk and then our waist and our hips and knees and legs and feet moving down the spinal column. So wherever that injury was determines what functions below that point are being compromised. So we look at different parts of the brain here. We have things that I want you to look up. And the cerebral cortex made up of 4 lobes. We look at the functions of the brain. You can go into that. And I end the chapter with sort of a story about my own experience in psychology. Some of you might know from having taken classes from me and getting my bio a number of times, is I started school as a marine biologist and studied that for a while. I really didn't like psychology, but the school that I went to had a Frisbee team, and I was on the Frisbee team, and the advisor for the Frisbee team was one of the psychologists. And he convinced me with my biological knowledge to take biopsychology. And we had a rat lab, and we ended up getting right into doing experiments that involved inserting electrodes into rats' brains. The sad part of that is because the rats' brains, we didn't have equipment to do any sort of x-rays to find out where our electrode truly went. You can see in the picture in the book here, this is I forgot the name of stereoscope or something like that. And it's these 2 bars are inserted into a live rat's ears. And they hold the skull in a particular orientation. And we perform the surgery, we take off part of the skull, and we insert the electrode into the brain, and we can measure impulses, or we can send an impulse and basically create a lesion or destroy part of that brain, and then observe the rat's behavior after that. Now, in order to specifically locate where that injury occurred, we actually have to kill the rat. So we kill the rat, we remove its head, we take all the tissue around from the brain, we freeze the brain, we rapidly freeze the brain, and slice it into very, very, very slim slices that can go onto slides. And that's the other picture that you see. And we can find the lesion, and then we correlate the behavior that we saw with the lesion. Now, got to tell you, I totally was into this. I was stoked about it. I took care of the rats. I loved the rats. They were all great, but they were experimental animals, doomed to die. And not quite sure how much of this is still going on. I mean, there's still animal studies that are used for drugs and for all kinds of parts of science, but it's certainly a controversial one when we start thinking about the experience of the animal. So going into our assessments for Chapter 4, the other things that you need to get done this week, we have a discussion about using animals in labs. Let's talk about that. We have a 1-question quiz where what is the advantage of the white matter in the spinal cord? And I said it today, so I said it right back. And just look that up. And then we have an assignment where what I want you to do is create a presentation. The presentation will have you put images together as if you were teaching somebody about the major parts of the anatomy of the nervous system. So go into some detail, but let's say maybe you're teaching another college student about what you're learning in biopsych. So let's see if you can get that done.